Welcome back to the deep dive. Today we're looking at something with two very different sides, environmental microbiology,
That's right. We're exploring how microbes are fundamental to life on Earth.
but also how they can pose these really serious threats, either naturally after disasters or, well, when used deliberately.
Exactly. It's a synthesis, really. On one hand, microbial ecology, how tiny organisms basically run the planet's nutrient cycles.
And on the other.
Public health preparedness, how those same microbes become critical factors in disease, especially when things go wrong, like infrastructure fails, or in bioterrorism scenarios.
Okay, so our mission today is to really connect those seemingly separate worlds for you. We're going from the absolute basics of the biosphere.
All the way to the details of, say, Category A bioweapons.
We want you to have those aha moments, understanding how fundamental life and global security are actually intertwined. Right, let's start with just the sheer scale. It's hard to grasp.
It really is. We tend to focus on visible life, plants and animals, but microorganisms, they make up roughly half of all the living biomass on the planet.
Half. Wow.
Yeah, they're the main engines driving all the nutrient cycles, the planet's economy, essentially.
And they don't live in isolation, right?
Right.
You mentioned communities.
Right, they form populations of the same species, then guilds, groups working together metabolically, and then diverse communities, all packed into tiny microhabitats within larger environments. They are everywhere, constantly working.
Okay, so let's talk about that work, the cycling of elements. Carbon first. That seems like the big one.
It is. Carbon is the backbone of, well, everything organic. The cycle itself is relatively straightforward compared to others.
So who's doing the primary work there, fixing the carbon?
That falls to the autotrophs, the self-feeders. We often think of plants, sure, but microbial photoautotrophs like cyanobacteria are huge players. They perform carbon fixation.
Taking CO2 out of the air and turning it into organic stuff using light.
Exactly. Then, when organisms like us, the chemoautotrophs, eat, or when decomposers break down dead things, that CO2 gets released back. It's a fundamental loop.
Okay, very simple. But then, nitrogen.
Nitrogen. Yeah, that's where it gets much more complex. The paradox is that nitrogen gas, N2, is almost 80% of our atmosphere.
But completely useless in that form to most life.
Precisely. It needs to be fixed. Nitrogen fixation is that crucial step, converting N2 gas into usable ammonia, NH3. And this relies on a powerful enzyme called nitrogenase.
And the tricky part.
Nitrogenase is incredibly sensitive to oxygen. It gets destroyed by it. So the microbes doing this fixing have evolved these amazing strategies to protect the enzyme.
Like what?
Well, some cyanobacteria have specialized cells called heterocysts that keep oxygen out. Others, like rhizobium, live symbiotically inside plant root nodules, where the plant helps maintain an oxygen-free zone.
Huh. It's like building a tiny, sealed-off factory just for that one process. Once it's fixed into ammonia, then what?
Then it goes through ammonification, breaking down organic matter, releases more ammonia, and then nitrification. That's a two-step process run by different microbial guilds.
Right. Ammonia to nitrites, then nitrates to nitrates, which plants can use.
Exactly. Nitrate is what agriculture often relies on, which leads to the flip side. Denitrification. This is where certain anaerobic microbes convert those valuable nitrates back into N2 gas.
And it just bubbles away, lost from the soil.
Yep. It's a significant economic loss in farming, a real headache.
Okay, but here's something really fascinating from the material, this anammox process.
Yes. Anaerobic ammonium oxidation. This was a relatively recent discovery, and it's pretty amazing. These bacteria take ammonium and nitrite and convert them directly into N2 gas without oxygen.
Directly. How significant is that?
It's huge. Estimates suggest it could be responsible for up to half of all the nitrogen gas produced in the oceans. And it offers a potentially much cheaper way to treat wastewater compared to traditional nitrification and nitrification.
But there's a catch.
There is. The process involves a toxic intermediate, hydrazine, rocket fuel, essentially. So while it works beautifully in nature, scaling it up safely for industrial wastewater treatment needs some serious bioengineering.
Wow. Okay, briefly, the other cycles. Sulfur and phosphorus?
Sulfur is a bit like nitrogen, but the main reservoir is in rocks and sediments, not the air. Microbes oxidize hydrogen sulfide, H2S, back to usable sulfate.
And phosphorus.
Phosphorus is unique. No major gaseous form. It cycles through rocks, water, and organisms, and tends to get locked up in marine sediments over long periods.
And this connects to a big environmental health issue, right?
Yeah.
Eutrophication.
Directly. That's what happens when you get excessive runoff of phosphates and nitrates, usually from fertilizers, into lakes or coastal waters.
It feeds these massive blooms of algae and cyanobacteria.
Exactly, an explosion of life. But then, as that bloom dies and decomposes, it consumes all the dissolved oxygen in the water.
Creating dead zones.
Right. It kills off fish and other aerobic life, and then anaerobic bacteria take over, often producing nasty stuff like hydrogen sulfide, that rotten egg smell. It's an ecological cascade triggered by nutrient pollution.
Okay, let's shift to where these microbes live. Soil, for instance. Teeming with life.
Absolutely teeming. Millions, even billions of bacteria per gram of topsoil. It's incredibly dense and diverse.
What controls who lives where in the soil?
Key factors are moisture essential for life oxygen levels. So waterlogged soil favors anaerobes. pH is important too. Bacteria generally like it neutral. Fungi can handle more acidic or alkaline conditions. And of course, temperature and the availability of nutrients like organic matter.
There's a visual tool for this, isn't there? The Winogradsky column.
Yes, it's a great way to visualize microbial communities and sediments. You set up a column with mud, water, and nutrients, and over time, different bacteria form distinct colored layers based on their needs for light, oxygen, or sulfur compounds. It's like a living demonstration of niche partitioning.
Fascinating. Now, aquatic environments. You mentioned the Lake Erie bloom.
A perfect and scary example. Microcystis aeruginosa, a cyanobacterium, produced microcystin, a potent liver toxin. It contaminated the water supply for Toledo, Ohio, impacting hundreds of thousands of people. Shows the direct public health link.
And lakes themselves have zones, vertically.
They do. Think of it in layers based on light and depth. Near the shore, you have the littoral zone. Lots of light, nutrients, rooted plants.
Then out in the open water.
That's the limnetic zone. Still well-lit surface water, dominated by phytoplankton and zooplankton.
Deeper down?
You hit the profundal zone. Light penetration is poor, oxygen levels drop. Organisms here mostly rely on organic matter drifting down from above. They're often called the benthos.
And the very bottom, the sediment.
That's the benthic zone. It's cold, dark, often low oxygen or anaerobic. You find organisms adapted to that, like various Clostridium species.
And in the ocean, there's even deeper.
Oh yes, the deep ocean has the vast abyssal zone. Down there, especially near hydrothermal vents, you find unique ecosystems based on chemoautotrophs using chemical energy, not sunlight. Some are even thermophiles, loving the heat.
Understanding these habitats and how they get disrupted seems like a good bridge to the next topic. Microbes and natural disasters.
It is, because the key concept here is that disasters don't usually introduce new diseases. What they do is dramatically increase the transmission of diseases already present or endemic in the population.
How does that happen?
Primarily through breakdown of infrastructure. Displacement forces people into crowded shelters, sanitation systems fail, clean water becomes scarce.
So floods and tsunamis are major culprits for waterborne diseases.
Absolutely. Fecal contamination of drinking water is the biggest risk. That leads to outbreaks of things like typhoid fever, cholera, hepatitis A. Direct contact with contaminated floodwater can also cause wound infections.
And the standing water itself?
Yeah.
Mosquito breeding grounds.
Exactly. That escalates the risk for vector-borne diseases like malaria, dengue fever, West Nile virus, depending on the region.
And you specifically mentioned leptospirosis.
Yes, that one has a strong link to floods. The bacteria are carried by rodents and shed in their urine. Floodwaters spread it widely in the water and mud. People get infected through cuts or sometimes just contact with contaminated water.
Okay, now this is interesting. The material addresses a common myth about disasters. The corpse myth.
Yes. This is important for anyone involved in response. There's this widespread fear that dead bodies after a disaster are a major source of epidemics.
But the evidence.
The evidence just isn't there. For the general public, corpses pose negligible risk of epidemics. The pathogens involved usually don't survive long in a deceased host or aren't easily transmitted that way.
So who is at risk?
The risk is primarily for workers who are handling the bodies routinely. They might face risks like TB from aerosols during handling, potential exposure to blood-borne viruses if they get injured, or gastrointestinal infections from contact with leaking fecal matter.
But for the population at large, the focus should remain on
clean water, sanitation, vector control, and managing the living displaced populations. Not the bodies themselves causing outbreaks. It's a crucial, counterintuitive point.
Right. Okay, let's pivot from natural threats to calculated ones. Bioterrorism. This isn't new, is it?
Not at all. Biological warfare goes way back. Think Assyrians poisoning wells or Tartars catapulting plague victims over city walls in the 14th century.
But the modern context is different. The 2001 anthrax attacks really highlighted the need for preparedness.
Absolutely. That event spurred the creation of structures like the CDC's Division of Bioterrorism Preparedness and Response. There's now a much more formalized system for assessing and responding to these threats.
And that system uses categories, A, B, and C.
Yes, it's a prioritization based on risk. Category A agents are the highest priority. They pose a significant risk to national security.
What makes something category A?
The criteria include things like, can it be easily spread or transmitted person to person? Does it cause high mortality? Does it have the potential to cause public panic and social disruption? And does it require special public health preparedness?
And this includes the big six: smallpox, anthrax.
Plague, botulism, tularemia, and the viral hemorrhagic fevers, yes.
Let's talk anthrax, Bacillus anthracis.
Uh-huh.
The inhalation form is the most feared for bioterrorism.
It is, because the spores are tiny, stable, and easily aerosolized. When inhaled, they get deep into the lungs, into the alveoli, and are taken up by immune cells to the lymph nodes in the chest.
And then they germinate.
Exactly. They switch from dormant spores to active vegetative bacteria, and then they start producing potent toxins. The key insight here is that the severity of inhalation anthrax is driven by the amount of toxin produced, not necessarily the number of bacteria present. It's the toxin that kills.
Right, what about botulism? That's caused by a toxin too, right?
Yeah.
Not the bacteria itself.
Correct. Clostridium botulinum produces the botulinum neurotoxin, which is the most potent natural toxin known. The potential lethality is staggering, theoretically. 1 gram could kill over 1 million people.
That sounds terrifyingly effective as a weapon.
It does, but it also has significant limitations.
Yeah.
The toxin itself is sensitive. It's denatured by heat. Proper cooking destroys it. Standard chlorination in municipal water supplies inactivates it. And if aerosolized outdoors, sunlight breaks it down relatively quickly within maybe 1 to 3 hours.
So those vulnerabilities are critical for defense.
Absolutely. Understanding how to neutralize the agent is key.
And the last group in category A, the viral hemorrhagic fevers, VHFs.
Yes. Caused by several different RNA virus families. Ebola and Marburg are filoviruses. Lassa is an arenavirus, and there are others. They cause severe, often fatal, multisystem illness with bleeding.
And handling them requires extreme precautions.
Definitely. Diagnosis and research require biosafety level 4, BSL-4 labs, the highest level of containment, because they are so dangerous and often lack effective treatments.
Okay, moving down a level, category B.
These are moderately easy to disseminate, cause moderate illness rates, and lower mortality than category A. Think agents like E. coli, O157, Toki7, Salmonella, Vibrio cholerae food and water safety threats primarily.
But you mentioned one toxin in this category.
Right. Staphylococcus enterotoxin B, SEB. It causes severe food poisoning symptoms. What makes it a persistent threat is that the toxin itself is very heat stable, so it can survive cooking or pasteurization processes that would kill the bacteria.
And category C.
These are emerging infectious disease threats that could potentially be engineered for mass dissemination in the future. Think things like Nipah virus or hantavirus. They have high potential, but maybe aren't easily produced or weaponized yet.
So faced with these threats, what's the role of healthcare and first responders?
Absolutely critical. Hospitals and clinics are on the front lines for recognizing unusual disease clusters, isolating patients, providing treatment. Clear protocols are essential decontamination procedures, knowing the chain of command for reporting suspected bioterrorism events to public health authorities like the CDC and potentially law enforcement like the FBI.
And there's a specific point about children's vulnerability.
Yes, the American Academy of Pediatrics pointed this out. Children are potentially more vulnerable to aerosolized agents. They breathe more air per pound of body weight. They're shorter, so closer to ground-level aerosols, and their skin is thinner and less keratinized. These factors need to be considered in preparedness planning.
And there's a network for lab testing.
Yes, the Laboratory Response Network, LRN. It's a nationwide network linking state and local public health labs with federal labs like the CDC. It ensures rapid capacity for testing and identification of biothreat agents.
We've really covered a huge range here, from the microbes that build the planet to those that could be used to attack it.
It's quite the spectrum. We've seen how microorganisms drive those essential biogeochemical cycles, how natural disasters can amplify the spread of existing diseases, and how specific microbes or their toxins pose calculated bioterrorism threats. Understanding all these facets is crucial, especially for healthcare professionals.
So let's nail down the 3 big takeaways for you. First, Microbes are the unseen powerhouses of the planet, running the critical nutrient cycles like carbon and especially the complex nitrogen cycle.
Second, natural disasters mainly escalate the transmission of diseases already present, often through water contamination. And importantly, the risk from corpses to the general public is actually very low, contrary to popular belief.
And third, defending against bioterrorism means knowing the specifics, the mechanisms of action, and the vulnerabilities of the high-priority Category A agents, like anthrax toxin and the botulinum neurotoxin.
Thinking about all this, it really highlights the power of microbial systems. We talked about the anammox bacteria and their potential for revolutionizing wastewater treatment. It makes you wonder. Well, considering how efficient these natural microbial processes can be, like anammox, compared to costly traditional methods, how else could we potentially leverage biotechnology and naturally occurring microbes to tackle other large-scale environmental or public health challenges? Beyond just nitrogen removal, what other problems could microbial solutions address?
That's a great question to leave everyone with. A lot to think about regarding the power hidden in the microbial world. Thank you for joining us on this deep dive today. We hope you found it insightful. We'll see you next time.